all patients with any stem cell loss were put into two groups, the two groups that I discussed, with stem cells and without stem cells. And everyone was randomized, which meant that no one knew which arm you were going into of the trial. And most of the patients with aniridia went into the trial arm, so they got stem cells. And most of the patients that had chemical burns went into the control arm. And it just happened that randomization resulted in that. But what that trial showed was that in patients that have aniridia and have the stem cell problems, by replacing the stem cells, it does have some impact. However, what we do know from other trials that have been done before, that over time, and certainly by 3 to 5 years, the stem cells that have been put on the eye from the surgery tend to disappear again, or they stop functioning again. And that leads us on to the new trial that Julie's hopefully going to be discussing with you in the next few talks. What we think happens in aniridia is that you can't just replace the stem cells, because the environment that the stem cells go in or stay in, that's abnormal as well. And historically, we haven't really replaced that. So what we plan to do is we plan to replace not only the stem cells by surgery, but also the environment that the cells are in as well. So by replacing everything, we hope that the outcomes will be much better. And that's a trial that's been funded by the Medical Research Council. We're hoping to start recruiting into the trial next year, probably middle to third quarter of next year. The other thing that we're looking at particularly is, so that's a surgical treatment, and that's what we can potentially offer and may well significantly help the cornea in patients who have the stem cell loss and aniridia. We won't know the outcomes for that for at least another 5 years beyond initiation of the trial. But we're also interested in knowing what we can do earlier on, so in the first and second decades of life. And we just started a project with one of our research fellows where we look after about 123 patients who have aniridia but have the corneal changes with it. So that's probably one of the largest groups of patients in the world. And what we're wanting to do is we've gone back and looked at what outcomes have happened from interventions that we've made, such as corneal transplants or cataract surgery. But we're also particularly keen on looking back at the mutations that everyone has. So a large proportion of patients who have aniridia will have mutation screens. They'll have had a blood sample taken away, and hopefully, you will also have had a sample taken. And we go and look to see what the mutation is. And why we're interested in that is we think that if you look at aniridia as a whole, not everyone gets the corneal changes, but some subgroup of patients do get it and get it very badly. We're interested in knowing what mutations particularly make you prone to that. And the reason for doing that is that we can then, when we have a child in front of us who has aniridia, we're able to then take their blood, do a mutation screen, and we're then able to determine whether or not they're at high risk of getting corneal problems. And if they are, we can then do much more frequent monitoring in the first and second decades of life. And there are certain things that we can potentially do that might delay the progression of the disease. For example, serum drops is one potential treatment which we don't often do early on. The other thing that we're wanting to look at is what particularly happens in those mutations, what other genes interact with that mutation, and then what the outcome is, and whether or not we can block those in some way. So that's really, hopefully, going to give you an idea of what we're currently doing with our aniridia research program. And there's obviously a lot of work that's still going on in the lab, but we're hoping, with the interactions that I have with Julie and her team, we're hoping to take this, a lot of those things into clinic much more effectively. I think that's probably about it. Alice,
Yeah, thank you, Saj. I mean, if anyone has any burning questions now for Saj, please feel free to raise your hand.
Just, you said about the environment for the stem cells. What do you mean by that? What was the actual?
So there's two. What we call stem cell deficiency is a disease where which happens from various causes, including aniridia. What happens is the white part of the eye, the conjunctiva, and its blood vessels come onto the cornea, and that happens for two main reasons. Either you've got a true loss of stem cells, so for example, if you've had a chemical burn, you tend to lose the number of stem cells. But there are many diseases, such as the inflammatory diseases and the genetic diseases like aniridia, where it's not just the fact that the stem cells are not normal in number. There might well be a normal number of cells, but they're not able to grow and proliferate on the eye. So what we call, they're not able to multiply on the eye to produce daughter cells. And what that then means is that if you normalize the environment, it could well be that the stem cells that are present there start functioning normally again. So stem cell deficiencies have historically been seen as just a loss of stem cells, but now we're sort of starting to realize that actually that's not the whole story. It's that you need to have the right number of stem cells, but you need to have the right environment, the right blood supply, the right growth factors in the environment, the right, what we call the matrix, so where the stem cells sit, that has to be normal.
So that would be what you'd add.
Yes.
What would how that work?
So whereas before we've just transplanted stem cells in the hope that that will increase the numbers, we will now attempt to transplant the stem cells with their own environment in place as well. So we normalise the environment. Does that answer your question?
Thank you.